T cells are a key part of the adaptive immune system. They fight viruses, they help keep cancer at bay, and they're also implicated in autoimmune diseases. But we still don't really know which T cells actually respond to which pathogens. I'm a PhD student at the Tri-Institutional Program in Computational Biology and Medicine in New York City, and I study this very problem of T cell specificity in the labs of Dr. Morris and Dr. Greenbaum. We have billions of T cells circulating in the human body. T cells are able to specifically recognize epitopes through their hypervariable T cell receptor, which I will be referring to as TCR. An epitope is a fragment of a protein that is presented on the surface of other cells, which could either be virus-infected or cancerous, and this protein from which an epitope is derived is then called an antigen. When a T cell recognizes an epitope that is different from the body's own proteins, the T cell activates and can target the infected cell. Why would we want to know which T cells recognize which antigens? Because it would have very wide clinical applications. Knowing T cell specificities can help diagnose and monitor infectious and autoimmune diseases. There is, for example, an FDA-approved T cell-based COVID-19 test in the market. It would also help us design more effective cell therapies like CAR T cells and predict whether a cancer patient would respond to immunotherapy. Until recently, the T cell specificity problem has been largely intractable because of the huge diversity of TCRs and potential epitopes and because of very little experimental data available. But over the last few years, several big data sets have been published, including a large functional assay-based dataset of paired TCRs and epitopes for COVID-19, which we're leveraging together with two collections of literature-sourced paired TCRs and epitopes from different antigen species. Our goal is to predict which TCR sequence corresponds to which epitope sequence. For this, my collaborator Artyom Streltsov and I built a model called BERTie, which is based on BERT, a transformer-based deep learning model which has been very successful in natural language processing tasks like machine translation. Transformers are also showing good results on biology tasks like understanding viral evolution, protein structure, and function. We overlay BERT with a multi-layer perceptron component, feed it paired TCR and epitope sequences, and a label which indicates whether this is a true or a fake pair, and train it to recognize true pairs. We experimented with training the model on the paired sequence prediction task directly, and also first explicitly pre-training it on a masked language model objective. In this step, the model would learn to complete masked out portions of the input sequences from context, and thus would learn an implicit representation of TCRs and epitopes. We also experimented with amino acid or character level and n-gram encodings. We benchmarked the model against two state-of-the-art methods, Ergo with autoencoder and Ergo based on LSTM. When trained and tested on exactly the same datasets, our different model setups consistently outperformed Ergo. We evaluated performance with ROC curve and the precision recall curve, which we think is more appropriate here because it evaluates predictions simultaneously at different thresholds. The best performing setup for BERTie was the 3-gram encoding model with the MLM pre-training, and in general this pre-training gave a slight lift to models in each encoding regime. We analyzed whether BERTie can help identify recognition motifs in the TCRs specific to a particular epitope. Here are two SARS-CoV-2 epitopes that were excluded from the training set. On the left side are logos for the TCRs that are reported by the functional assay as recognizing those two epitopes, and on the right-hand side are the top TCRs predicted by BERTie to be specific for these epitopes, and we can see that BERTie does uncover what looks like the recognition motifs. We then wanted to see whether BERTie can identify clinically relevant TCRs in actual patient TCR repertoires. Here's an example of a patient at our hospital that developed COVID-19 with TCR sequencing done at different points during the disease. This plot shows frequencies of the TCRs that are known to be COVID-associated through the functional assays. We also see other large TCR clones that display similar expansion dynamics expected during active infection. So we believe that these are also COVID-19 associated TCRs. Here are TCRs that the best performing Ergo model predicts as TCRs recognizing COVID-19 epitopes. As you can see, it identifies two possibly relevant TCRs. The best performing BERTie identifies a bigger proportion of these relevant TCRs. Here's an example of another patient, Ergo trained with additional features such as the type of T cell which would not be available in practice, does better in this case than plain Ergo with sequences alone, but BERTie, trained on the TCR and epitope sequences alone, still uncovers more of these potential COVID-19 associated TCRs. We are continuing our work on further improving the model and validating whether it will generalize from the largely virus epitope setting to cancer neoantigens. For example, whether we can correctly predict TCRs that recognize the recurring TP53 mutations in cancer patients that have responded to immunotherapy. We're also diving into the model introspection to extract recognition motifs and hopefully draw conclusions as to how TCRs generally recognize antigens. I'd like to thank my supervisors, Quaid Morris and Benjamin Greenbaum, my mentor, Yuval El-Hanati, and my two very supportive and fun labs, our sources of funding, and my PhD program. Thank you for your interest, and I hope to see you at the poster session.